Hey everyone, welcome to this explainer. If you're a ninth grader gearing up for that New York State Biology Regents exam, and yeah, I know you're probably feeling that study anxiety starting to creep in right about now. Well, take a deep breath, because you are in the exact right place. Today, we're going to demystify genetics, and more importantly, we're going to uncover the ultimate Regent study hacks, straight from the official standards together. We're looking at the true blueprint to the building of life, and we're going to make sure you are perfectly prepared.
Okay, so here's our roadmap for today. We've got 5 stops. 1. The instruction manual, which is DNA. 2. Building the body through mitosis. 3. Mixing it up with variation. 4. Predicting traits using probability. And finally, 5. Passing it on through reproduction. All right, let's jump right into section 1, the instruction manual, DNA. So let's start with the absolute foundation. You probably already know that DNA and chromosomes act as this fundamental instruction manual, right? They pass characteristic traits from parents to offspring. But what does the Regents actually care about here? Well, they want you to understand the difference between coding and non-coding DNA. It's kind of like this. Coding Regents, those provide the actual readable instructions for your traits. They're the literal sentences in the manual telling your cells exactly what to do. You know, giving you brown eyes or making sure your body produces the right enzymes. Now, non-coding regions are simply the other parts of the DNA sequence. Think of them like the blank margins of a page, or maybe the spacing between paragraphs in a textbook. They don't give the direct instructions for building a specific trait, but they are absolutely essential for organizing the book so it can actually be read properly. Understanding this specific distinction is a direct Regents requirement, so definitely lock this concept in.
Moving right along, we hit section 2, building the body, mitosis. Now, when our bodies actually read that manual to grow, they use cellular division, or mitosis. And we really need to grasp the difference between normal and abnormal division here. When normal mitosis happens, it leads to healthy growth, development, and stem cell differentiation. And hey, differentiation is really just a fancy way of saying cells are figuring out what specific job they're going to do in your body, like a blank slate stem cell getting the signal to become a specialized skin cell, or a beating heart cell, or a neuron in your brain. It is an incredibly precise process. But when this highly regulated process breaks down, we get abnormal division. This leads to unregulated, totally out of control cell growth, and the primary example of this is cancer. The test really wants us to understand these specific big picture outcomes rather than getting lost in the weeds. Which brings us to a massive, crucial point. Wait for it. The assessment boundary actually says, do not memorize the specific steps of mitosis. Yes, you heard me right. This is our first major study hack directly from the official test standards. You do not need to stress over the specific biochemical steps, no prophase, metaphase, anaphase, and no memorizing specific gene control mechanisms. The state just wants you to know the outcomes we literally just talked about: healthy growth, stem cell differentiation, and what happens when it goes wrong, like cancer. So, take a deep breath and scratch those complex cellular steps right off your study guide.
Next up is section 3, mixing it up, variation.
Let's shift gears a bit. If we all have DNA, how do we end up with such unique, individualized instruction manuals? Well, that comes down to inheritable genetic variations. And there are 4 key sources of variation you need to be able to defend with evidence. First up is meiosis, which creates entirely new genetic combinations when reproductive cells form. This is exactly why you might look like a mix of your parents, but aren't an exact clone of either one of them. Second, replication errors, specifically viable errors during copying. Now, I always think viable errors sounds like a terrible indie rock band name, but in biology, it just means a tiny typo in the DNA copying process that the organism actually survives and passes on. Pretty cool, right? Third, we have environmental factors. These can cause mutations from your surroundings, like UV radiation from the sun affecting your skin cells. And fourth is genetic engineering, which represents human-driven advances in biotechnology where scientists directly modify DNA. So those 4 factors, that's exactly how unique traits are created and passed on.
And guess what? Here is another massive relief for us. Skip the specific phases of meiosis. Focus on the data. Just like with mitosis, the Regents assessment boundary explicitly states they are not going to ask for the specific biochemical phases of meiosis. You don't need to draw out the complex steps of reproductive cell division. Why? Because honestly, the state cares way more about the macro outcomes than the micro mechanics. Your focus needs to be on how these processes, along with environmental factors and biotechnology, actually lead to variation in the real world. So keep your eyes on the big picture here.
All right, let's jump into section 4, predicting traits, probability.
So let's take this to the next step and see how it all builds. How often do these unique genetic remixes actually show up?
We're moving our explainer from the microscopic level of individual cells all the way up to the whole population level. When we look at a hypothetical probability distribution of expressed traits in a population, we see that it's influenced by multiple factors, specifically genetic factors and environmental factors interacting together. The test wants you to understand that we can use statistics and probability to predict these traits. It's not just about what genes are there. It's about how the environment interacts with those genes. For instance, your genes might give you the biological potential to be very tall, but environmental factors like childhood nutrition actually interact with those genes to determine your final physical height. It's the combination of the two that determines what traits actually get expressed in a group of organisms over time.
But wait, if we're talking about statistics and probability at the population level, does that mean complex math? Boom, hack number 3, 0 Hardy-Weinberg calculations on the test. Let's just celebrate this massive relief together for a second. The assessment boundary explicitly states that those complex, multivariable Hardy-Weinberg math equations will absolutely not be on the exam. No way. You just need to understand the overarching concept of probability and how traits are distributed without ever having to crunch the advanced algebra. Finally, we reach our last stop, Section 5, passing it on, reproduction.
We've reached the final, macro-level stage of our biological journey, how organisms pass their manual to the next generation to maintain the continuity of life. Following the timeline, fertilization kicks the whole thing off, leading right into embryonic development, where the organism actually begins to form and grow. Now, during this incredibly sensitive time, developing embryos interact with other human body systems and are deeply, deeply affected by environmental influences. For example, a mother's diet, her overall health, or exposure to toxins in the environment can directly impact how that embryo develops. All of these interacting factors ultimately lead to the continuity of life. It's this beautiful, complex cycle focusing on how the structures and functions of human reproductive systems sustain our entire species.
To round out our explainer today, here are your final stress-relieving study hacks of what not to study. Do not memorize the specific details of hormonal regulation. And do not memorize the specific stages of embryonic development. Seriously, cross them right off your review checklist. Understanding the general function, the interactions between body systems, and the impact of the environment, that is what's going to score you the points on the Regents. You really don't need to know the names of every single hormone or the exact day-by-day cell count of an embryo.
So, we have gone all the way from the microscopic letters of DNA to the macro continuity of human life. But I want to leave you with a thought that goes a bit beyond the textbook today. With genetic engineering and stem cell research advancing at literal lightning speed, how will humans shape our own continuity of life in the future? It's a massive, provocative question. And as the next generation of scientists and informed citizens, you'll be the ones to eventually answer it. Thank you so much for joining me for this explainer. We uncovered the blueprint, we found the assessment boundaries, and you are absolutely going to find incredible success on your Regents exam. Keep learning, keep questioning, and I'll catch you next time.